Welcome to Lecture 17 of Biology 116 entitled Development 2. In this next lecture on development, what we're going to be focusing on is essentially where we left off from the previous lecture on development, and that was basically at the blastula level. What we have done in the previous lecture was look at fertilization in great detail, sperm meeting egg, and then we've looked at some of the cleavage events that occur in order for that zygote to eventually turn into a developing embryo at the stage that we know of, which would be the blastula. Now, in this lecture, what we're going to do is essentially go all the way from the blastula stage to the moment of birth of an organism. So it's a very big jump, very broad jump, lots of stuff has to go on. Thus, this is going to be a very content-heavy lecture, as we'll see. So let's begin with an overview of what we're going to be looking at throughout this lecture. There's going to be two main themes of this lecture, and those two themes are going to be centered around two processes that I'll put on the side here for right now. And that would be gastrulation, which is something we've seen before, and also a process called organogenesis. So we of course know that this term, this root of genesis, means birth, and here we're going to give birth to the organs of an organism, because if something is born, it is of course born with all of its organs, and that's something we have to do embryonically in terms of development, and we also have to undergo this process of gastrulation. More on that a little bit later. So let's take an overview of what this lecture is going to be about. First of all, what we have to remember and sort of recall is cleavage as a process. That was a lot of development 1, and that was devoted to looking at cleavage patterns and influences of cleavage and what that eventually results in. Basically, if you remember from development 1, cleavage is basically going to be when cells divide. But specifically, cells are dividing very rapidly. That's the big thing. Cells divided very, very rapidly. So rapidly, in fact, during their rapid growth, during their rapid, division, what these cells did is that they had no time for growth. So there was no time for any cell growth. And overall, that resulted in what? That overall resulted in cells being very, very small. All of the cells in this developing embryo were very, very small, since they never grew, since they never underwent G1 and G2. They sort of skipped those phases of the cell cycle just to make sure that they continuously did the S&M phases, synthesis and mitosis, over and over and over again. Remember, cleavage is cell division without growth, and that's essentially what happened in the previous lecture. But now what we want to do is talk about what happens once cleavage is done. We're now obviously going to have to grow as an organism. We can't stay at the small blastula level. We have to grow. So eventually what we're going to see in this lecture is the following. Eventually, you're going to have to resume normal cell cycle. You can no longer skip the G1 and G2 phases as we did in the cleavage stage of life. Right now we're going to resume the normal cell cycle. What is that going to lead to? That will essentially lead to time for something very important in embryonic development, and that's growth. Essentially, when we are doing growth, we are then going to be having a normal cell cycle process for this growth to even occur. And again, how is this growth happening? Why didn't this happen before? Well, that was because the rate of cell division was so fast. What we're going to do now, essentially in this lecture, is slow it down. So the rate of cell division slows down, and the direct consequence of slowing down the rate of cell division is increasing the amount of growth within the cells. Rate of cell division slows down. Okay, so that's sort of one half of this lecture that we'll see today. And also what we're going to see is another process that results in this, organogenesis. And that would be a process known as morpho, which refers to shape, genesis. We're going to give birth to the shape of the organism. Morphogenesis, more broadly speaking, could just be the change of shape in an organism. And specifically, this change of shape that we're focusing on, this morpho that we're focusing on, is at the embryonic level of development. So we'll say of an organism. Specifically, we'll now sort of put some more to this. We're going to basically be looking at both cellular plus tissue processes that result in this. And these are going to be at the both cellular and tissue level, of course. And these are things that we'll see in this lecture. Overall, when you change the cellular morph, and you change the tissue morph and shape, you get the animal body to take a true shape. So the animal body will then find its true sort of birth form, birth shape, will take shape. So we know how what an infant looks like. It has a characteristic human shape, right? This is because of morphogenesis, because of cellular and tissue-based processes resulting in the taking of a true sort of animal shape, whatever the shape may be. And in addition, morphogenesis is going to require two sort of processes to occur as well. So it occurs during the last two stages of embryonic development. And that's something we sort of highlighted and alluded to before, but now we're just going to explicitly mention that during embryonic development, the two stages of focus in this lecture that is going to result in the fully formed infant, let's say, that we're trying to get to, will be gastrulation and, of course, organogenesis. And when you combine those two processes, you will eventually end up with a fully embryonically developed organism that will be birthed, that will leave the home of the mother and now will be in the open world. And that's what we're going to be looking at throughout this lecture.